And this would be the part of the mRNA that codes for our gene attenuator peptide. This would be the CPA1 gene. And as its elongation is occurring, amino acids are linked together, forming our gene attenuator peptide. However, in the presence of arginine, stalling, degradation of mRNA, and that's how the regulation works. Importantly, it's domain specific, so our gene attenuator peptide is native in yeast, and it only functions in eukaryotic ribosomal systems. Similarly, prokaryotic examples of stalling sequences only work in prokaryotic ribosomes. So as I mentioned, ribosomal stalling is dependent on what that sequence is, but can also be co-induced with a small molecule, like arginine. This is medically significant because sometime in the future there's potential for drugs to be developed that can, say, downregulate the expression of a disease protein. So to try to learn more about the mechanism of ribosomal stalling and what's really happening, we look to the ribosome structure to see what possible interactions between the nascent chain or the nascent protein are having with the ribosome exit tunnel. This is a diagram of the large subunit and the exit tunnel I mentioned earlier. These three gray blobs are proteins that help make up the ribosome. Notably, there's a peptidyltransferase center where amino or peptide bonds are formed, and the growing polypeptide extends through here, having to traverse a constriction of the exit tunnel. So L4 and L17 form this narrowing that the growing polypeptide has to go through. And this is cryo-EM, so basically electron density kind of showing that. This would be our gene attenuator peptide. Importantly, there is data from another lab, their paper, showing highly ordered density, which was suggested to be our gene attenuator peptide, this yellow blob, in contact with a loop of L17 and L4 at the constriction site, as well as with L10 in the peptidyltransferase center. So this density suggests that, there might be some interaction at these locations. So what I proposed was that our gene attenuator peptide-induced stalling is dependent on these specific areas of the protein. Meaning, if, say, I mutate these small parts of the proteins, then stalling should not persist. And that would help narrow down what kind of interactions are going on, which proteins are involved in what parts of those proteins. So the goal was basically to create a ribosome, say, that had a mutant, as I mentioned, near the constriction, like for L17, for example, out of the 3 proteins I looked at. It's a little difficult. There's multiple copies in different places of the yeast genome, and they're very essential and highly expressed because ribosomes are very abundant in cells, and they're sensitive to mutation. How I did this was introducing a plasmid or a circular piece of DNA to kind of reprogram the expression of the cell. What happens is with these specific strains from the milkweed lab, or this part was from the milkweed lab basically, we introduced this. If you grow the cells in glucose, then the wild type is highly repressed, and our mutants can be, the cells have to function off our mutant copies. So the first step was figuring out what mutations can the yeast ribosome tolerate. Out of the time I had this summer, I found 2 that worked and several that didn't. So far I have an E. coli eyes L4 and E. coli eyes L7. The colors here don't really match, but just as I was trying to explain earlier, what I did was change the sequence so that it's normal except for the loop, say, near the constriction is with E. coli, which, since our gene attenuator peptide only stalls in eukaryotic systems, it's essentially a less severe deletion of those loops. Because of the high selection pressure, it was necessary for me to double check the amino acid sequence of the plasmid I introduced to make sure no recombination events had occurred. This is just a readout from the sequencing, and it looked good, even with the high selective pressure when grown in glucose. So we checked the amino acid sequence of the plasmid. The next was to check if the expression levels actually followed the repression under glucose of the wild type. So I did a reverse transcriptase quantitative PCR to analyze this. Basically what happens is there's mRNA that the cell is making to make all its proteins, which then gets reverse transcribed into DNA, and then polymerase chain reaction into a lot of pieces of DNA. So the more mRNA you have at the beginning, which correlates with more expression, then the faster this exponential growth in the polymerase chain reaction is going to happen. So I have a plot of my L17 data. Basically, the orange lines are the control, and this is switching from the dark lines are galactose sugar, and the light lines are glucose. So if you grow the cells in glucose, then they're forced to live on the, hopefully on the mutants. And that's what this showed. The wild type went down, went to the right, which means expression was downregulated. And for our mutants, it moved to the left. So that looks good. So we have our mutant, has the right sequence, has the right expression potentially, or it seemed like it had the right expression. The next step was to create cell-free translation extracts. Basically, for eukaryotic systems, I wanted to be able to have our in vitro solution of our mutant ribosomes and other components necessary for translation, program that with the arginine attenuator peptide mRNA, and then monitor stalling. So I did. The first prep, I guess I got kind of lucky. L4 less efficient than L17, but still enough of a background to test if stalling occurred. So as I said, I needed to program these translational lysates with mRNA. Basically, arginine attenuator peptide ahead of luciferase. Luciferase is a gene from fireflies that fluoresces. So if there's more expression, then there's more fluorescence, and that's how you measure it. So the ideal thing we'd see is if we increase the arginine concentration sufficiently, then expression should decrease because of stalling, which is what happened in our wild type. So that was the control, the positive control. It worked. L4, because the ribosomes were less efficient and the mRNA was less efficient, it wasn't enough, but we got L17 data so far. The E. coli is L17. So as I mentioned, the wild type expression levels went down by about half, so that means that stalling occurred. However, in the L17 mutants, stalling did not occur. So this supports my hypothesis that this specific L17 loop is necessary and vital for stalling. And it suggests that the constriction site is part of the mechanism of ribosomal stalling, at least for arginine attenuator peptide. Future directions definitely involve getting the other mutants up to speed that I started on. And for example, the L4 wasn't efficient enough, getting that increased efficiency. Possibly using the yeast arginine attenuator peptide sequence instead of Neurosporapassa homolog, which is also a type of fungus. And controlling for endogenous amino acids, because since stalling is dependent on the concentration of arginine, that's pretty relevant. There could be stalling could be happening more than we know about. Definitely knocking out the wild type genes would kind of, the RTQ PCR I did, that would for sure mean that they're living off the mutant ribosomes. And then even further down the line, we're piecing down which part of the ribosome is interacting with the nascent chain, and figuring out how that's communicated to the rest of the ribosome to elicit elongation arrest. So I'd just like to thank my postdoc mentor, Nathaniel Littner, worked with me very closely with this, and he was always helpful. Professor Jamie Cate, the entire Cate lab, Nathan and Leah from CERT, and of course the Rose Hills Foundation. Thank you.